Thanks, Allison. Thanks for the organizers for the opportunity to be here and share some of the things that we've been working on. I apologize, I probably have too many slides. I'm going to move kind of fast, but I just kind of want to give you a flavor of the things that we think about. We're very interested in how to program mechanical function of DNA origami. This is a little bit outdated picture of our group. Just quick conflict of interest disclosure. We co-founder and recently started a startup company. But just to give you a sense of what mechanical functions we're really interested in, we're interested in things like actuating conformational changes, generating forces, higher order assembly, and in particular also adaptive structure and properties of these higher order assemblies. And we think about basing those on kind of underlying design foundation of DNA origami, but how do we design the structure, mechanical properties, dynamic properties, and functionalization, and sort of bring that all together into these more complex mechanical systems. And so we try to apply these mechanical functions in different areas, right? Nanorobotics, tools for biology is becoming increasing of interest, and then biomedical nanodevices. I won't really talk about biomedical nanodevices, but I'll show you quickly two examples kind of spanning these tools for biology and nanorobotic devices and materials. All right, and again, trying to tie these to that underlying design foundation. Right, if we think about trying to build nanocalipers to mechanically probe biomolecules, I highlight some of the underlying design challenges and goals for those tools on the right side. And then later, we'll see if we have time to get into these reconfigurable adaptive assemblies. So of course, we use DNA origami. I probably don't need to spend much time on this, but basically programming folding of this well-known scaffold sequence into a higher order structure by designing the sequence of these staples to pinch these together into higher order structures. And this is really the baseline, one example of how we're trying to take that DNA origami and use it as a mechanical tool to interact with biomolecules. The basic idea is illustrated here, right? But down at the bottom, you'll see some of the characteristics that we'd like to achieve with these tools. One is measure nanoscale distances, measure conformational changes, apply forces, and measure mechanical response. Right, and then get towards implementing those in a more realistic biological setting. And again, kind of tying that to how we think about designing those features into these tools. There's a lot of work in this area developing tools. I'm going to, in the interest of time, right, we don't really have time to go through a comprehensive review. I'll just highlight some of the work that we've done. All right, we typically start with thinking about how do we design the motion here. I had taken this slide out, but I put it back in. All right, it didn't serve anyway, I wanted to show you that we were often kind of like the last talk, right, thinking about how do we take inspiration from macroscopic machines to sort of scaffold our design process. This is supposed to show a video of this robot moving around, and we kind of try to approach our DNA-based nanoscale design, a similar approach of how we design kinematic motion. But the basic idea of how we design motion is we take these rigid parts, fit them together in a more complex geometry, and we facilitate that motion through flexible domains, particular single-stranded DNA. Right, and we're able to achieve either rotational motion. Right, hopefully these videos are not playing. Okay, anyway, you should see a video of these hinges opening and closing and these sliders extending and retracting. Sorry, I think something got messed up in the upload. But so, we've been working a lot on motion for many years, and in the last few years, we've been really trying to take that now to things like force application, right, or mechanical responses. And so we're trying to understand how do we design the mechanical properties of these structures. We've been inspired by a couple of computational studies. This in particular one is from the Doi lab, looking at some of the hinges we previously designed. All right, and so this guided us to think about controlling mechanical properties, right? We have these stiff domains here, these stiff arms connected by single-stranded DNA, and they showed that you can tune the mechanical properties by controlling the base paired states of that single-stranded DNA. All right, so we developed this new version of a caliper where we have very long single-stranded DNA regions on the back end, and the idea is we have some baseline, pretty flexible hinge, right, and then we can control the mechanical properties of that hinge by how we base pair that single-stranded DNA on the back end. Right, and without going into the details, we've been able to show we have these design approaches to kind of tune these free energy landscapes, or basically tune the mechanical properties. Right, that allows us to think about designing these tools that can apply different forces to different size molecules in this range of either a few piconewtons in compression or a few piconewtons in tension. We've gotten interested in applying these tools to study chromatin through collaboration. And so, again, in the interest of time, I won't go through the details, but we're interested in particular these chromatin subunits, these sets of 4 nucleosomes, because it is a biologically relevant substructure, and the dynamics of that control accessibility of DNA. All right, and just to quickly highlight, we have been able to show that with these tools, we can apply tension to these tetranucleosome structures, chromatin substructures, and actually use that to drive decompaction of these structures. So you can see here 1 dot and 2 dots are compacted structures, and as we apply tension with this caliper tool, we can actually drive decompaction, right, and that allows us to study some of the physical properties of these chromatin substructures. All right, so we are able to design, use structure and dynamic and mechanical design to develop these tools where you can measure distances, measure conformational changes, apply forces, and measure a mechanical response. And one of the things we're actively working towards is implementing those in real biological systems. And then just in the last another minute or so, I wanted to show you kind of one of the things we're very interested in, becoming increasingly interested in these sort of nanorobotic materials, is having collections of many dynamic devices that we build up into larger systems to achieve some kind of coordinated function. This is kind of the simplest example where we have basically assembly and displacement and sort of adaptive mechanical properties where we can assemble this hinge into different kinds of structures. There's many nice examples of taking similar DNA origami subunits and creating different larger assemblies. Again, we're particularly interested in, can we make those dynamic and mechanically functional? And so we've been collaborating with Nick Stephanopoulos' group to use this coil-based peptide assembly and basically combining that with these dynamic hinge devices, and we can make different kinds of interesting higher-order assemblies. You can see these are micron or a few microns big, and then we can get micron scale distance changes and actually also do things like control the mechanical properties. So if we actuate these hinges, we can get differences in the overall persistence length, the overall stiffness of these polymers by a factor of about a 2.5 to 10-fold increase in stiffness by actuation. And so again, just kind of wrapping up quickly, these are the areas we're interested in, these nanorobotic devices and materials, kind of leveraging these tools to make these probes that we can study biology with, and then interest in biomedical that I'm happy to talk about offline. But that's all kind of based on this underlying foundation of thinking about how do we design the structure, the mechanical properties, dynamic properties, and functionalization of these DNA devices. And so with that, I'll end, thank the funding, and happy to take any questions that people might have.
Wonderful. Thank you so much. We have about time for 1 or 2 questions before we ask how it fits into the architecture.
Can you just expand very slightly on the nanoscale robotics, like what the robots would actually be doing, or the robotic, or was it robotic material?
Yeah, so in general, right, we get thinking inspiration from macroscopic engineering. Robotic materials are materials that sort of sense and respond to their environment, have adaptive properties, so maybe color change, right, or think of things that might sense the environment and camouflage, right? We're interested in trying to build some of those fundamental capabilities of sensing. I didn't show, we have some other projects focused on signal transmission, sort of distributed coordinated behavior, so maybe a local signal create an overall shape change of this micron scale assembly, so things in that direction.
What do you think is the lowest hanging fruit for a robotic material?
I think there could be interesting applications in things like maybe there's folks here that work on things like artificial muscle. We're really interested in sort of, can we embed these into synthetic cellular systems, maybe things that reconfigure to drive long-range cell-cell interactions, or even things like, things that clamp onto a cell and drive exocytosis or endocytosis with these conformational changes. So can we couple those conformational changes onto other material systems?
Okay, cool. Well, then now I have our question, which is, how does it actually fit into one of the architectures? Do you have any ideas?
Sure. Yeah. So I mean, we are interested. Obviously, there's nice examples of these 3D printers and things like that. We've thought about things in that direction. Also, scaffolding materials over a larger scale. Right. So we've thought about maybe not building molecules, but building nanoparticle assemblies. Right, where you have kind of similar. We're good at building these large, big things that move sort of tens of nanometer to micron scale, and then maybe using that to build organized assemblies of nanoparticles that can exhibit interesting optical or electronic properties.
Wonderful. Any question to that or comment or addition? Yes.
What's the highest level that you can get rigidity to? How tall, how big can you get?
I mean, it depends. What rigidity is kind of a tricky word, and it means it's always a question of how rigid. But we can make things that are, let's say persistence lengths of tens of microns, 20 microns. So you can build very, very stiff thing, but then it's always a balance of building something super stiff, but then we also want to build in dynamics that we can reconfigure. So I think that's the big challenge. You can make something very, very stiff over 10 micron scale, but then having something that is reconfigurable and stiff is a challenge.